constituents of extracellular and intracellular fluids. Textbooks. Guyton and Hall. Textbook of Medical Physiology. 11th edition. Pages 469 to 482. Red blood cells, erythrocytes. The major function of red blood cells, also known as erythrocytes, is to transport hemoglobin, which in turn carries oxygen from the lungs to the tissues. In some lower animals, hemoglobin circulates as free protein in the plasma, not enclosed in red blood cells. When it is free in the plasma of the human being, about 3% of it leaks through the capillary membrane into the tissue spaces or through the glomerular membrane of the kidney into the glomerular filtrate each time the blood passes through the capillaries. Therefore, for hemoglobin to remain in the human bloodstream, it must exist inside red blood cells. The red blood cells contain a large quantity of carbonic anhydrase, an enzyme that catalyzes the reversible reaction between carbon dioxide, CO2, and water to form carbonic acid, H2CO3, increasing the rate of this reaction several thousand-fold. The rapidity of this reaction makes it possible for the water of the blood to transport enormous quantities of CO2 in the form of bicarbonate ion, HCO3, from the tissues to the lungs, where it is reconverted to CO2 and expelled into the atmosphere as a body waste product. The hemoglobin in the cells is an excellent acid-base buffer, as is true of most proteins, so that the red blood cells are responsible for most of the acid-base buffering power of whole blood. Shape and size of red blood cells. Normal red blood cells, shown in figure, are biconcave discs having a mean diameter of about 7.8 micrometers and a thickness of 2.5 micrometers at the thickest point and 1 micrometer or less in the center. The average volume of the red blood cell is 90 to 95 cubic micrometers. The shapes of red blood cells can change remarkably as the cells squeeze through capillaries. Actually, the red blood cell is a bag that can be deformed into almost any shape. Furthermore, because the normal cell has a great excess of cell membrane for the quantity of material inside, deformation does not stretch the membrane greatly and, consequently, does not rupture the cell, as would be the case with many other cells. Concentration of red blood cells in the blood. In normal men, the average number of red blood cells per cubic millimeter is 5,200,000, plus or minus 300,000. In normal women, it is 4,700,000, plus or minus 300,000. Persons living at high altitudes have greater numbers of red blood cells. This is discussed later. Quantity of hemoglobin in the cells. Red blood cells have the ability to concentrate hemoglobin in the cell at up to about 34 grams in each 100 milliliters of cells. The concentration does not rise above this value, because this is the metabolic limit of the cell's hemoglobin-forming mechanism. The concentration does not rise above this value, because this is the metabolic limit of the cell's hemoglobin-forming mechanism. Furthermore, in normal people the percentage of hemoglobin is almost always near the maximum in each cell. However, when hemoglobin formation is deficient, the percentage of hemoglobin in the cells may fall considerably below this value, and the volume of the red cell may also decrease because of diminished hemoglobin to LL the cell. When the hematocrit, the percentage of blood that is cells normally, 40 to 45%, and the quantity of hemoglobin in each respective cell are normal, the whole blood of men contains an average of 15 grams of hemoglobin per 100 milliliters of cells. For women, it contains an average of 14 grams per 100 milliliters. Each gram of pure hemoglobin is capable of combining with 1.34 milliliters of oxygen. Therefore, in a normal man, a maximum of about 20 milliliters of oxygen can be carried in combination with hemoglobin in each 100 milliliters of blood, and in a normal woman, 19 milliliters of oxygen can be carried. Production of red blood cells. Areas of the body that produce red blood cells. In the early weeks of embryonic life, primitive, nucleated red blood cells are produced in the yolk sac. During the middle trimester of gestation, the liver is the main organ for production of red blood cells, but reasonable numbers are also produced in the spleen and lymph nodes. Then, during the last month or so of gestation and after birth, red blood cells are produced exclusively in the bone marrow. Relative rates of red blood cell production in the bone marrow of different bones at different ages. As demonstrated in figure, The bone marrow of essentially all bones produces red blood cells until a person is 5 years old. The marrow of the long bones, except for the proximal portions of the humeri and tibiae, becomes quite fatty and produces no more red blood cells after about age 20 years. Beyond this age, most red cells continue to be produced in the marrow of the membranous bones, such as the vertebrae, sternum ribs, and ilia. Even in these bones, the marrow becomes less productive as age increases. Genesis of blood cells. Pluripotential hematopoietic stem cells, growth inducers, and differentiation inducers. The blood cells begin their lives in the bone marrow from a single type of cell called the pluripotential hematopoietic stem cell, from which all the cells of the circulating blood are eventually derived. Figure shows the successive divisions of the pluripotential cells to form the different circulating blood cells. As these cells reproduce, a small portion of them remains exactly like the original pluripotential cells and is retained in the bone marrow to maintain a supply of these, although their numbers diminish with age. Most of the reproduced cells, however, differentiate to form the other cell types shown to the right in figure. The intermediate stage cells are very much like the pluripotential stem cells, even though they have already become committed to a particular line of cells and are called committed stem cells. The different committed stem cells, when grown in culture, will produce colonies of specific types of blood cells. A committed stem cell that produces erythrocytes is called a colony-forming unit, erythrocyte, and the abbreviation CFUE is used to designate this type of stem cell. Likewise, colony-forming units that form granulocytes and monocytes have the designation CFUGM, and so forth. Let's say it again. A committed stem cell that produces erythrocytes is called a colony-forming unit, erythrocyte, and the abbreviation CFUE is used to designate this type of stem cell. Likewise, colony-forming units that form granulocytes and monocytes have the designation CFU, GM, and so forth. Growth and reproduction of the different stem cells are controlled by multiple proteins called growth inducers. 4 major growth inducers have been described, each having different characteristics. Point 1 of these, interleukin 3, promotes growth and reproduction of virtually all the different types of committed stem cells, whereas the others induce growth of only specific types of cells. The growth inducers promote growth but not differentiation of the cells. This is the function of another set of proteins called differentiation inducers. Each of these causes one type of committed stem cell to differentiate one or more steps toward an adult blood cell. Formation of the growth inducers and differentiation inducers is itself controlled by factors outside the bone marrow. For instance, in the case of red blood cells, exposure of the blood to low oxygen for a long time results in growth induction, differentiation, and production of greatly increased numbers of erythrocytes. In the case of some of the white blood cells, infectious diseases cause growth, differentiation, and eventual formation of specific types of white blood cells that are needed to combat each infection. Stages of differentiation of red blood cells. The RST cell that can be identified as belonging to the red blood cell series is the proerythroblast, shown at the starting point in figure. Under appropriate stimulation, large numbers of these cells are formed from the CFUE stem cells. Once the proerythroblast has been formed, it divides multiple times, eventually forming many mature red blood cells. The RST generation cells are called basophil erythroblasts because they stain with basic dyes. The cell at this time has accumulated very little hemoglobin. In the succeeding generations, as shown in figure, the cells become LLED with hemoglobin to a concentration of about 34%, or the nucleus condenses to a small size, and its null remnant is absorbed or extruded from the cell. At the same time, the endoplasmic reticulum is also reabsorbed. The cell at this stage is called a reticulocyte because it still contains a small amount of basophilic material, consisting of remnants of the Golgi apparatus, mitochondria, and a few other cytoplasmic organelles. During this reticulocyte stage, the cells pass from the bone marrow into the blood capillaries by diapedesis, squeezing through the pores of the capillary membrane. The remaining basophilic material in the reticulocyte normally disappears within 1 to 2 days, and the cell is then a mature erythrocyte. Because of the short life of the reticulocytes, their concentration among all the red cells of the blood is normally slightly less than 1%. Function of the erythropoietin mechanism to increase production of red blood cells when tissue oxygenation decreases. Regulation of red blood cell production. Role of erythropoietin. The total mass of red blood cells in the circulatory system is regulated within narrow limits so that 1. An adequate number of red cells is always available to provide sufficient transport of oxygen from the lungs to the tissues, yet 2. The cells do not become so numerous that they impede blood flow. What we know about this control mechanism is diagrammed in figure and is as follows. Tissue oxygenation is the most essential regulator of red blood cell production. Any condition that causes the quantity of oxygen transported to the tissues to decrease ordinarily increases the rate of red blood cell production. Thus, when a person becomes extremely anemic as a result of hemorrhage or any other condition, the bone marrow immediately begins to produce large quantities of red blood cells. Also, destruction of major portions of the bone marrow by any means, especially by X-ray therapy, causes hyperplasia of the remaining bone marrow, thereby attempting to supply the demand for red blood cells in the body. At very high altitudes, where the quantity of oxygen in the air is greatly decreased, insufficient oxygen is transported to the tissues, and red cell production is greatly increased. In this case, it is not the concentration of red blood cells in the blood that controls red cell production but the amount of oxygen transported to the tissues in relation to tissue demand for oxygen. Various diseases of the circulation that cause decreased blood flow through the peripheral vessels, and particularly those that cause failure of oxygen absorption by the blood as it passes through the lungs, can also increase the rate of red cell production. This is especially apparent in prolonged cardiac failure and in many lung diseases, because the tissue hypoxia resulting from these conditions increases red cell production, with a resultant increase in hematocrit and usually total blood volume as well. Erythropoietin stimulates red cell production, and its formation increases in response to hypoxia. The principal stimulus for red blood cell production in low oxygen states is a circulating hormone called erythropoietin, a glycoprotein with a molecular weight of about 34,000. In the absence of erythropoietin, hypoxia has little or no effect in stimulating red blood cell production. But when the erythropoietin system is functional, hypoxia causes a marked increase in erythropoietin production, and the erythropoietin in turn enhances red blood cell production until the hypoxia is relieved. Role of the kidneys in formation of erythropoietin. In the normal person, about 90% of all erythropoietin is formed in the kidneys, the remainder is formed mainly in the liver. It is not known exactly where in the kidneys the erythropoietin is formed. One likely possibility is that the renal tubular epithelial cells secrete the erythropoietin, because anemic blood is unable to deliver enough oxygen from the peritubular capillaries to the highly oxygen-consuming tubular cells, thus stimulating erythropoietin production. At times, hypoxia in other parts of the body, but not in the kidneys, stimulates kidney erythropoietin secretion, which suggests that there might be some non-renal sensor that sends an additional signal to the kidneys to produce this hormone. In particular, both norepinephrine and epinephrine and several of the prostaglandins stimulate erythropoietin production. When both kidneys are removed from a person or when the kidneys are destroyed by renal disease, the person invariably becomes very anemic because the 10% of the normal erythropoietin formed in other tissues, mainly in the liver, is sufficient to cause only 1/3 to 1/2 the red blood cell formation needed by the body. Effect of erythropoietin in erythrogenesis. When an animal or a person is placed in an atmosphere of low oxygen, erythropoietin begins to be formed within minutes to hours and it reaches maximum production within 24 hours. Yet almost no new red blood cells appear in the circulating blood until about 5 days later. From this fact, as well as other studies, it has been determined that the important effect of erythropoietin is to stimulate the production of proerythroblasts from hematopoietic stem cells in the bone marrow. In addition, once the proerythroblasts are formed, the erythropoietin causes these cells to pass more rapidly through the different erythroblastic stages than they normally do, further speeding up the production of new red blood cells. The rapid production of cells continues as long as the person remains in a low oxygen state or until enough red blood cells have been produced to carry adequate amounts of oxygen to the tissues despite the low oxygen. At this time the rate of erythropoietin production decreases to a level that will maintain the required number of red cells but not in excess. In the absence of erythropoietin, few red blood cells are formed by the bone marrow. At the other extreme, When large quantities of erythropoietin are formed available and if there is plenty of iron and other required nutrients available, the rate of red blood cell production can rise to perhaps 10 or more times normal. Therefore, the erythropoietin mechanism for controlling red blood cell production is a powerful one. Maturation of red blood cells requirement for vitamin B12, cyanocobalamin, and folic acid. Because of the continuing need to replenish red blood cells, the erythropoietic cells of the bone marrow are among the most rapidly growing and reproducing cells in the entire body. Therefore, as would be expected, their maturation and rate of production are affected greatly by a person's nutritional status. Especially important for normal maturation of the red blood cells are two vitamins, vitamin B12 and folic acid. Both of these are essential for the synthesis of DNA, because each in a different way is required for the formation of thymidine triphosphate, one of the essential building blocks of DNA. Therefore, lack of either vitamin B12 or folic acid causes abnormal and diminished DNA and, consequently, failure of nuclear maturation and cell division. Furthermore, the erythroblastic cells of the bone marrow, in addition to failing to proliferate rapidly, produce mainly larger than normal red cells called macrocytes, and the cell itself has a MZ membrane and is often irregular, large, and oval instead of the usual biconcave disc. These poorly formed cells, after entering the circulating blood, are capable of carrying oxygen normally, but their fragility causes them to have a short life, 1/2 to 1/3 normal. Therefore, it is said that deficiency of either vitamin B12 or folic acid causes maturation failure in the process of erythropoiesis. Lifespan and destruction of red blood cells. When red blood cells are delivered from the bone marrow into the circulatory system, they normally circulate an average of 120 days before being destroyed. Even though mature red cells do not have a nucleus, mitochondria, or endoplasmic reticulum, they do have cytoplasmic enzymes that are capable of metabolizing glucose and forming small amounts of adenosine triphosphate. These enzymes also, 1. Maintain pliability of the cell membrane, 2. Maintain membrane transport of ions, 3. Keep the iron of the cell's hemoglobin in the ferrous form rather than ferric form, and 4. Prevent oxidation of the proteins in the red cells. Even so, the metabolic systems of old red cells become progressively less active, and the cells become more and more fragile, presumably because their life processes wear out. Once the red cell membrane becomes fragile, the cell ruptures during passage through some tight spot of the circulation. Many of the red cells self-destruct in the spleen, where they squeeze through the red pulp of the spleen. There the spaces between the structural trabeculae of the red pulp, through which most of the cells must pass, are only 3 micrometers wide, in comparison with the 8 micrometer diameter of the red cell. When the spleen is removed, the number of old abnormal red cells circulating in the blood increases considerably. Destruction of hemoglobin. When red blood cells burst and release their hemoglobin, the hemoglobin is phagocytized almost immediately by macrophages in many parts of the body, but especially by the Kupffer cells of the liver and macrophages of the spleen and bone marrow. During the next few hours to days, the macrophages release iron from the hemoglobin and pass it back into the blood, to be carried by transferrin either to the bone marrow for the production of new red blood cells or to the liver and other tissues for storage in the form of ferritin. The porphyrin portion of the hemoglobin molecule is converted by the macrophages, through a series of stages, into the bile pigment bilirubin, which is released into the blood and later removed from the body by secretion through the liver into the bile.